Okay, so today we're going to cover SARS, ACE2, and spike proteins. Um, SARS in the context of SARS-CoV-2, ACE2 in the context of the protein that it binds in order to gain entry into cells and infect them, and spike proteins in the context of the new mRNA injections. In order to understand these principal items, you have to first understand the renin-angiotensin system. So I'm going to give some background on that. And once we understand that, I'm going to discuss the SARS-CoV-2 involvement and also the potential mRNA spike protein involvement. So most people want to know why and how SARS-CoV-2 in particular is bad for some people. We hear a lot of talk about comorbidities in the elderly, but there might be something else going on here because young people are suffering severe pathologies as well. And there is speak of long COVID as well. So just as a preface to what I'm about to describe, the renin-angiotensin-aldosterone system is a system that every single human being has, and it may be very intimately tied into the pathogenesis of COVID-19 viral illness. Inadequate ACE function, and I'll get to what that is an independent predictor of mortality. So I want to talk about the renin-angiotensin-aldosterone system first, and I'm going to do a very slow and comprehensive description of what this system is. And again, this is a system that has nothing to do with SARS. It's a system that exists in every single person, and it's essential to the functioning of blood pressure and an electrolyte balance. So the Raas system for short, renin-angiotensin-aldosterone system, is a closed-loop system in everybody, and its principal job is to regulate blood pressure and also to regulate fluid and electrolyte balance. This is primarily done by the kidneys. There are many organ systems involved, and I'll go through them now quickly. The kidneys are very important in this system, and they're responsible for excretion of waste, blood ion regulation, blood pH regulation, hormone production, and blood volume regulation. And as I said, they play a very important role in the Raas system, which I'll get to in just a moment. The liver is also involved in this closed-loop system. And the liver, for those of you who don't know, is used to filter blood, detoxify, synthesize and store amino acids, proteins, vitamins, and fats. And it also regulates blood glucose, and it functions to drain bile. Also involved in the closed-loop system are the lungs, and of course everybody knows that the lungs are involved in oxygen exchange and distribution, and for ventilation and diffusion, and they of course are intimately connected to the heart, comprise the cardiovascular system, and of course the heart is also an organ that's involved in the Raas system. And our amazing hearts pump for the entirety of our lives oxygen-rich blood throughout our bodies and keep us alive. So these are the principal organs involved in the system. So now I'm going to describe how they're involved and interconnected and how this closed-loop system works. So we have a model of a human being here with all their organs exposed. And we have an artery shown on the right, on the top. And we also have the adrenal glands, which sit on top of the kidneys, shown on the bottom right. So this just contextualizes where all the organs are. So the kidneys sit behind. You can't really see them here, but they're very important in this system. When the kidneys are compromised due to for example, a systemic drop in blood pressure, or say the artery feeding the blood to the kidney is compromised somehow, they respond to this stress by producing a protein called renin. Actually, there's a protein called prorenin, which is already circulating. So the signal sent by the kidneys converts it to renin. The liver, which is shown here in the organ systems, is responsible for the production of the hormone called angiotensinogen. And angiotensinogen is paired with renin, the protein which is produced by the kidneys, and it is converted to another hormone called angiotensin 1. Almost simultaneously to the production of this hormone is a very essential component in this system called ACE, which is an enzyme produced by the lungs. And ACE's function, angiotensin converting enzyme, is to convert angiotensin 1 to angiotensin 2, as you'll see coming up in the next slide. Angiotensin 2, once converted by ACE, is in its active form. So this is the form whereby it can do its function, act in two primary places. First of all, it acts on arteries, and second of all, it acts on adrenal glands, these glands that sit on top of the kidneys. So its function with its action on the arteries is to cause the arteries to constrict, which naturally raises blood pressure. Its function on the adrenal glands is to cause the adrenals to produce another hormone called aldosterone. And aldosterone acts in turn, closing this loop, on the kidneys, and the combination of the rise in blood pressure and the presence of aldosterone causes the kidneys to take up more salt and water from the urine, and thus the system starts to regulate itself and it stops producing so much renin. Now, what's the function of ACE2 in all this? Because this is the very receptor that our SARS virus binds. So this principal component in this system is very important for us to talk about. So its role, it's a membrane-bound protein, so it sits in the membrane of cells, and it binds and it acts on angiotensin 2, this hormone that we just talked about that acts on arteries and adrenal glands. So what ACE2 does is it converts angiotensin 2, this active form of this hormone, into another hormone called angiotensin 1-7. And angiotensin 1-7 is extremely important in closing this loop system, in that it also acts on arteries and it acts on the adrenals, much like angiotensin 2, but it does kind of the opposite action. So instead of causing the blood vessels or the arteries to constrict, it causes them to dilate. So what this does is it causes the blood pressure to regulate itself that way. And it also acts on the adrenals, as I said. And the way that it acts on them is to cause the adrenals to produce less aldosterone. So the kidneys end up getting a lower signal from the adrenals to subsequently keep the cycle going. So this is part of the closed loop system. So all of these components are required. Now, ACE2 is a special membrane-bound protein in that there's another protein or enzyme called shuttled, which can cleave or cut the external portion of this membrane-bound protein to allow the external portions to become free. So this is free or soluble angiotensin ACE2, angiotensin converting enzyme 2. So these free ACE2s can also act on the angiotensin 2 in order to convert it to angiotensin 1,7. And the effect, of course, is going to be the same. Angiotensin 1,7 will subsequently act on the arteries and on the adrenal glands, and subsequently start to close the loop of the Raas system. So it's an ebb and flow of a number of proteins and hormones that regulates this system, which ends up regulating blood pressure by vasoconstriction and dilation, and also the regulation of the salt in the water, the electrolyte levels. So all of these organs are required, and all of these organs that are required have to be functioning properly in order for the system to work. If you've been paying attention, you'll notice that there are two places in the system where the SARS-CoV-2 virus can potentially interfere. Now, we all know, or we should know by now, that the ligand, the binding, the protein that the SARS spike protein likes to bind when it enters the human body is the ACE2 membrane-bound protein, or the ACE2 protein. So it can do so, and it can also bind the free ACE2. These are actual crystal structures, and basically that means that a lab or a bunch of labs did a lot of work to crystallize, to purify these proteins and show what they look like using X-ray crystallography. So these are actually the way that the spike proteins are folded when they are either bound to their cognate ligand, the ACE2, or when they are unbound. The point is that there are two scenarios where they can bind. And the first one is designed by design, so the ACE2 can, so the spike protein, which is attached to the virus, can enter the cell by binding first to ACE2. The second scenario is that the free ACE2 can also bind the spike protein of the SARS virus. So if you're starting to wonder if there's going to be some kind of pathology associated with that, then you're probably thinking on the right track. So the occupation of the binding site, or the induction of a conformational change, may prevent angiotensin II binding. So think of it as a block. You have your SARS virus with your spike protein on its surface, and it's bound to free ACE2. Subsequently, ACE2 won't be able to do its action. It won't be able to bind or act on enzymatically angiotensin 2 such that it gets converted to angiotensin 1,7. So it's like a block. So the normal functioning Raas system would be interrupted by the presence of, let's say, large numbers of SARS virions. So to summarize this part of the idea in the presentation, competitive inhibition by blocking binding of angiotensin 2 with ACE2 in order for ACE2 to complete its function as an enzyme to convert angiotensin 2 to angiotensin 1,7 may result in an overactive Raas system, or it may actually infuriate an existing overactive Raas system, which, of course, we all know results in many pathologies such as hypertensive disorders. So the bottom line here is that it can interfere both at the transmembrane level and also at the free ACE2 level to potentially interfere with the Raas system. So we're going to jump into the next section, which is on the subject of the injectables, the mRNA products being produced by Moderna and Pfizer. So in this last part, we'll cover what mRNA is and how these mRNAs are being delivered as injections and what the mechanism of action is meant to be. So mRNA is basically coding material. Coding material in living organisms is in the form of DNA or RNA, pardon me. And this coding material gets transcribed into messenger RNA, which is basically a template for the construction of proteins via another process called translation. So the reason it's called translation is because you go from a sequence of nucleotides, which are the principal components of your coding material, into a sequence of amino acids. So I won't go into the details here, you don't need to know that, but when you hear people talking about an mRNA quote-unquote vaccine, the mRNA is the coding material for a protein. So this is a system in biological entities that exists. The way that we make the building blocks of our existence via making proteins is by converting the coding material in the form of an mRNA template into a protein. So this is natural. Our bodies do this, our cells do this via ribosomes, etc. So mRNA is the template. It's a coding sequence for a specific protein. And the specific protein that we're talking about here is the spike protein, the pre-fusion conformation of the spike protein, which looks like a triple-pronged pointy thing. So, as I was saying, it's delivered intramuscularly in a nano-lipid particle solution directly into your cells. So, the lipid nanoparticles are actually designed by a biotech company, and they're approximately 100 nanometers across using the technique that you can see in the laminar flow mixing schematic here on the right. It's about the same size as a SARS virus, actually, and these nanolipid particles envelop the mRNAs, they encapsulate them, and this is by design, because mRNA is pretty fragile, and you can't just inject foreign mRNA into cells because it will just degrade and nothing will happen. So you have to send them in the form of a package. So a nano-lipid particle solution is the packaging that these companies have decided would be the best route. So these packages are taken up, so-called, by our antigen-presenting cells, and they are processed. And subsequently, along the processing line within the host cell, the mRNA, the template, is dumped and eventually makes it to the processing plant, which is the ribosome. Again, I'm not going to go too deep into this. But safe to say, the host cells, the machinery of the host cells, produce large quantities of these proteins from these mRNA templates. And then they're digested and presented on special molecules called MHC molecules for presentation to T cells and B cells. And not only that, but the spike proteins themselves, the pre-fusion version of the spike proteins, are also presented on the surface for presentation to B cells. And in addition to that, the spike protein, the pre-fusion version of the spike proteins, are emitted from the cells. So that in turn means that these proteins are released into the blood, into the plasma, as free proteins. So we have spike proteins digested in little bits on the surface of MHC class molecules, presentation to T cells. We have presentation on the surface as a full structure, and we have exogenous spike proteins, which is just external to the cells floating around in the periphery. And this is where problems might start. If we have the spike protein in solution and we have free ACE2, which we will have as part of the renin-angiotensin system functioning, then these two can bind each other because they actually have a very high affinity bond, these two proteins, which means not only do they bind efficiently, they stay bound. So if this is the case, then we have competitive inhibition here, which means that the free ACE2 that are meant to be acting on the angiotensin 2 are not, because they're bound. And that either blocks or causes a conformational change in the protein itself so that it can't do what it's meant to do, which is bind to the angiotensin 2 and convert it to angiotensin 1,7. So it's quite easy to see how it's possible that this could very well happen, and if it did happen, of course it would be worse in someone who already has an overactive Raas, or you can also imagine it inducing an overactive Raas, and these are of course associated with hypertension, kidney problems, liver problems, coronary problems. There are a lot of things associated with an overactive Raas, which has been studied out the yin-yang. So the spike proteins that are produced by the host cells from the mRNA template, from the injection, may interfere with the renin-angiotensin-aldosterone system. That's the take-home message here. It needs to be studied intensely, as a matter of fact. So to answer the question, why is it bad for some people, and why is it worse for other people? The answer, in my opinion, would be, if you already have an overactive Raas system, which many people do, especially the elderly, then throwing a wrench in this works is going to make everything worse. So today I covered the renin-angiotensin system, I hope adequately, and SARS-CoV-2 involvement by binding ACE2. And also I talked a little bit about the potential mRNA spike protein involvement, which is quite concerning. many more researchers need to get on this and do some bench work to find out if this is in fact the case. So I have a few references here, some papers that people can read if they're interested. Somebody actually did some great work to show that eusia and anosmia, which is the lack of or the loss of taste and what's the other one? Anyway, the lack of sensory ability has been associated with the Raas, with the dysfunction in Raas. So there are many adverse events being reported right now as part of the vaccine adverse event reporting system in the US, which I also have done a study on recently, that involves many events that could very well be signs of a dysfunctional Raas system. So this also needs to be examined. So that's about it. I'm going to conclude because I don't want to take up too much time. I promised this would be about a half an hour. And I hope that it's been informative. And if you have any questions, you can drop them in the YouTube link at the bottom. Thanks for listening.